We're excited to have our next speaker here today. Our next speaker is Dr. Thomas Seyfried. He is a professor of biology at Boston College. He has received his Ph.D. in Genetics and Biochemistry from the University of Illinois. He holds a master's degree in genetics from Illinois State University. He was a Postdoctoral Fellow in the Department of Neurology at the Yale University School of Medicine, and then served as faculty as an assistant professor of neurology. Dr. Seyfried previously served as chair, scientific advisory committee member for the National Tay-Sachs and Allied Disease Association. He has received a Lifetime Achievement Award for his work on cancer from the Academy of Complementary and Integrative Medicine. He presently serves on several editorial boards, including those for Nutrition and Metabolism, Neurochemical Research, the Journal of Lipid Research, and ASN Neuro. Dr. Seyfried has over 180 peer-reviewed publications, and he is the author of the book Cancer as a Metabolic Disease. Dr. Seyfried served with distinction in the United States Army in the 1st Cavalry Division during the Vietnam War. He has received numerous medals and commendations. Please join me in welcoming Dr. Thomas Seyfried.
I want to speak to you today about how we view the origin of cancer, some of the problems, and then talk about how we can move forward with effective therapies based on a reevaluation of what this disease actually is. So I like to start off with kind of a report card, if you will. How are we doing in our attempts to manage this disease? So this is just over the last 5 years. I have data that go back to 1990, but if you just look at the data, these are from data from the American Cancer Society. So from 2013 to 2017, new cases, deaths per year, deaths per day. Not good, all right? So how would we rate our progress in managing cancer in the United States? I would say F, all right? In fact, the rate of increase for deaths per year is now surpassing, to some extent, the rate of new cases. So we have to say to ourselves, 1,600 people a day are dying from cancer in the United States. I just came back from China. They have about 8,000 to 9,000. In fact, it's overtaken heart disease in China, cancer. It's all over the world. Increasing deaths. So we have to ask ourselves, what's going on here? We run for this type of cancer. We swim for that type. We're raising money for cancer all the time. The NIH is increasing budgets for cancer. Pink ribbons, all this kind of stuff. Kids running, biking, swimming, raising money for cancer research. And what do we get? More cancer. How could this happen? How is it possible that we can't significantly drop the death rate for this disease? And that has to do with how we view the origin of the disease. And this is, in my view, the big problem. Is it a genetic disease, as we've been told, or is it a mitochondrial metabolic disease that I'll tell you about? This is a simple cartoon of a cell. So you see the nucleus in the center, and the little bean-shaped organelle is the mitochondria, and you get the plasma membrane. Okay, cancer cells are screwed up in so many different ways, right? We know we have mutations in the nucleus. There's no denying that. Although we know of a number of cancers that have no mutations in the nucleus. Very few people talk about that. And then we have damage in the mitochondria. And the mitochondria is the organelle that's shown in the little bean shape there in the orthodox model. It controls the energy of the cell. Both of these organelles are screwed up together with a variety of other organelles inside that cell. So what's the origin of the disease? The present dogma says it's the genetic mutations that are causing this disease. And I'll show you test evidence that say that's not the case. And this is what I think is the crux of the problem. I think this is what the problem is. So, and as I put these slides up and keep saying this over and over again, cancer is a genetic disease according to the dogmatic view. And this Hallmarks of Cancer by Doug Hanahan and Bob Weinberg pretty much solidifies this whole process. There's a number of other big papers saying the same thing. Cancer cells carry the oncogenic and tumor suppressor mutations that define cancer as a genetic disease. All right, so this is on the NCI website. Go on the NCI website, National Cancer Institute, cancer is a genetic disease. Go into any college textbook, biology, biochemistry, cell biology, cancer is a genetic disease. This has now become a dogmatic view, an irrefutable truth. No one discusses any other alternative except cancer is a genetic disease. And this dogma has now indoctrinated several generations of physicians and scientists into this worldview of what this disease is. 1,600 people a day are dying. Now, here's a cartoon from one of the biology textbooks to introduce the young student into the nature of what we're talking about. We got a car that's speeding out of control with no brakes. To illustrate the concept of tumor suppressor genes and oncogenes. So the brakes are like tumor suppressor genes. The car has no brakes. The cell has no brakes. This car is speeding. The oncogenes are forcing this group of cells. So cell division out of control is the phenotype of what we're talking about. All right, the signature issue of cancer, cell division out of control, driven by oncogenes and defective tumor suppressor genes. Accumulation of random mutations, the development of cancer cell. So we have a normal cell, and then we have, on the far right, we would have the malignant cell. And you can see it's dysmorphic. How many mutations cause cancer? Nobody knows. All right, this is not determined. 1, 2, 4. You know, some tumors have thousands and thousands. This guy says there might be millions of tumors. But Sir Michael Stratton. And they spend enormous amounts of energy and effort trying to find all the mutations in these tumor cells, and nobody really knows what's going on with this stuff. Now, the end result, what we have is the result of this dogmatic view of what the nature of cancer is personalized therapy, personalized medicine. You can call it what you want. It's the new thing. Personalized therapy based on cancer mutations and our attempt to either diagnose the disease or potentially manage the disease. You see this woman staring into the screen, looking at a bunch of spots, looking with so-called mutations in breast cancer. Maybe can help the diagnosis, or $7,200 to run a scan on your tumor cells to see what mutations might be there. Now, this would be all well and good if that information were really helpful, and whether or not the mutations meant anything. All right. So if the cancer is not a genetic disease and these mutations are downstream epiphenomena, then this activity is a complete waste of time and money. So when you try to tell people that's not what they thought it was, a dogmatic view is often, when you try to change dogma and you show them evidence, they don't want to look at it, they don't want to talk about it, they don't want to hear about it. Because it's challenging. Dogma is a powerful effect on the human brain. It can be religion, it can be politics, or it can be a scientific dogmatic view. So what I had done, I don't have time to go through all the research present here, I'll summarize it. But basically I plucked observations out of the literature over decades that had been published in various peer-reviewed journals and put them all together in this one paper, evaluating when you take the nucleus of a tumor cell and put it into a cytoplasm of a normal cell, and then if you take the normal nucleus and put it into a cytoplasm of a tumor cell, what do you get? And how do you interpret those data based on the two theories? Cancer is a mitochondrial metabolic disease, cancer is a nuclear genetic disease. How do you interpret the results from those experiments? And these experiments were done on all kinds of different models of cancer, done in different systems by different investigators. Most of the investigators were premier leaders in the field. And they didn't interpret their results in the light of the two competing theories. So it was kind of ambiguous at best. So now that I've identified this, I allow individuals themselves to go and read this paper. Don't listen to what anybody else tells you. If you're interested in this subject, you will read that paper and you will come to your own decision as how you view those data. And that's what I say. Let people read it for themselves. So what I've done, and my students, this is simply a cartoon that summarizes a lot of what was in those papers in a very simplistic way. Green cells beget green cells, that's normal cells. Red cells beget red cells. These are tumor cells. Tumor cells beget other tumor cells. Where do the tumor cells come from? Where's this dysregulated growth coming from? Is it driven by the mutations in the nucleus, as the dogma would have us believe? Or is it defects in the cytoplasm and the mitochondria, which are also defective in these cells? So the experiments were done very clearly in multiple different systems. You take the red nucleus, plucked it out of the cytoplasm of the tumor cell, and you put it into the cytoplasm of the normal cell, and you got normal cells, sometimes normal tissues, sometimes a whole mouse or frog cloned from the nucleus of a tumor cell. Not dysregulated cell growth that you would have expected if the driver genes were actually driving the phenotype. On the other hand, like Israel and Seyfried did, they took the nucleus out of the normal green cell and put it into the cytoplasm of the tumor cell, and they either got dead cells or tumor cells. They never got normal cells. And when Benny Kirpatu from Baylor College of Medicine switched the mitochondria, and he was able to show that if you put green mitochondria in a red cell, they convert the cell to a normal growth pattern. Completely opposite of what the gene theory would have said. All right. So these data clearly indicate that somatic mutations alone cannot be responsible for the origin of this disease or driving the phenotype of the disease, which is dysregulated cell growth. These are the strongest data to date that disprove the gene theory. Well, if that's the case, what are we doing? Right? I don't want to hear about this. I don't want to talk about it. I don't want to look at it. Okay, somatic mutations are not the origin of cancer. How do we get cancer? Well, Otto Warburg had this defined quite a while ago, back in the 1920s and 30s. Cancer arises from damage to cellular respiration. Okay, I can't breathe. How am I going to live? Fermentation gradually compensates for insufficient respiration. Cancer cells continue to ferment lactic acid fermentation in the presence of oxygen, which is the Warburg effect. You shouldn't be fermenting in the presence of oxygen because that would be a normal Pasteur effect. Cancer cells have an abnormal Pasteur effect. They continue to ferment in the presence of oxygen. What Warburg did not know, and that we are now working on, we're able to show that cancer cells not only can ferment carbohydrate or glucose, they also ferment glutamine, which he did not know about, which is, in my view, the missing link in his whole theory. And we'll be able to show that shortly. Enhanced fermentation is the signature metabolic malady of all cancer cells. Right? If you look at the genetics of any cancer cell, it's going to be different. Every cell in that tumor is going to have a different genetic profile of mutations, but they're all fermenting. So why are we chasing the mutations when they differ from every cell, every tumor, and all of them are fermenting? Doesn't it make sense to target the common pathophysiological problem of these cells than chasing all these secondary effects? 1,600 people a day are dying. Okay, look at this. This is a very simplistic overview of energy metabolism. We teach this to our freshman students at Boston College who are not even biology majors. We breathe air. We get most of our energy through oxidative phosphorylation. 80 to 90% of the energy we get comes from oxphos and generated ATP. We get much less oxygen from glycolytic pathway in the cytoplasm or in the succinyl-CoA ligase step in the Krebs cycle. So we get very little, that's called substrate level phosphor. It's a non-oxidative phosphor, a little bit in the cytoplasm, a little bit in the Krebs cycle, TCA cycle. Most of our energy comes from respiration, oxygen. What cancer cells are doing is they're generating more and more of their energy from substrate level phosphorylation, which allows them to live in hypoxic environments, and less through electron transport chain. So when we reconfigure the facts of disease in light of a mitochondrial metabolic disease, we can explain the majority of observations that have been found over the decades in this disease. The oncogenic paradox has perplexed cancer researchers and scientists for decades. It was first pointed out by Albert St. Georgi back in 1979. And what it basically says is, how is it possible that any kind of a plethora of provocative agents from the environment or from inheritance, and I'll talk about that, could cause cancer through a common pathophysiological mechanism that wasn't understood? In fact, if you read Sid Mukherjee's book, The Emperor of All Maladies, Pulitzer Prize-winning book, was on the bestseller list in the New York Times for over a year. He struggled with the oncogenic paradigm. Check page 285 and 303 of Sid's book, and you'll see we don't understand how this is all happening. Every one of those provocative agents that I point out there does one thing. It damages respiration. It produces reactive oxygen species. And we've heard some of that earlier today. Reactive oxygen species can be good when they're in low, but could be bad if they're too high. And what happens is the mitochondria becomes defective and they start blowing out reactive oxygen species, which are carcinogenic and mutagenic, therefore causing the point mutations and the chromosomal defects that you see in the nucleus are coming indirectly as the result of the ROS. The ROS are also further damaging the mitochondria, then signaling the mitochondria suffocating now. It's not producing enough energy. Well, if it doesn't produce enough energy, it's going to die. How are these cells going to survive if they can't get energy through oxphos? They better be able to ferment. And that's what the bottom showing here. Oxidative phosphorylation goes down, substrate level phosphorylation goes up. The mitochondria signal the nucleus. How am I going to get an inefficient energy source into this cell to keep its proliferative capacity or to keep it alive? And therefore, oncogenes turn on, which are transcription factors that upregulate to transporters to bring in glucose and glutamine into that cell. So what we have seen as being causative are actually an effect of trying to keep the cell alive with a non-oxidative fermentation process. So carcinogens, radiation, hypoxia, inflammation, rare inherited mutations, BRCA1, P53, they will only cause cancer if they in fact damage the mitochondria. If they don't damage the mitochondria, you don't get cancer, even though you might have that mutation. Therefore, they're secondary, not primary. Viruses, age, I went through all of that. So then what happens, you get the warbird. The cells start to begin to ferment. So the first three hallmarks of cancer, insufficient regulation, growth out of control, are simply the cell falling back on an ancient metabolic program that existed before oxygen came onto the planet some 2.5 billion years ago, when everything on the planet was a fermenter. Okay? So they're all fermenting. And what do? They ferment any kind of a molecule that you could find in the microenvironment. When the fermentation fuel ran out, the cell would die. So we're seeing that. Now, this is the strategy that we're going to take to manage this. So they enter their default state. Then you could get, now what happens? These cells ferment in our body. They throw out metabolized molecules like lactic acid, succinic acid, carbonic acid, all these different things acidifying the microenvironment, leading to an anti-angiogenic effect, into an angiogenic effect. All right, so this is all explained. Okay, so now apoptosis. Cancer cells avoid programmed cell death. How is that possible? Who controls the life of our cells? The mitochondria. They are the apoptotic signal. They are the kill switch of our cell. If the kill switch is broken, this thing is gone. Right? It doesn't have control. So the mitochondria that actually control the life of our cell and tells the cell it should be dead now is broken. So this cell continues to survive and is surviving on fermentation. How do we get metastasis? This is the big killer of most people, metastasis spreading from origin. So if the metastasis doesn't kill you, the treatments they use to treat it sometimes will kill you. Okay, so how do we get metastasis? Okay, so here we have a bunch of normal cells, columnar epithelial. They become dysmorphic. Okay, through one of the provocative agents in the microenvironment. Those red cells are now our immune system cells. They come in to heal the wound. They're wound healing cells. All right, they throw out cytokines and growth factors to try to heal the wound. This is wrong. So they're doing what they're genetically programmed to do in a wrong context. Now that fuels the cells to grow even faster and more malignant. Macrophages are also highly fusogenic. If they can't, they can actually fuse with other cells to facilitate wound healing. Now, what happens is these cells are now diluting their mitochondria in the cytoplasm, becoming more and more destabilized. So what eventually emerges is a macrophage with abnormal mitochondria. So this macrophage is already genetically programmed to enter and exit the bloodstream, to live in hypoxic environments. This is the toughest cell in our body, and now it's rogue, and it's running around, and it's very hard to kill. No new mutations are not necessary. Also, they live in hypoxic environments. So what does that say? All this anti-angiogenic therapies are not going to work. Avastin, crap. All right? Spending millions of dollars on anti-angiogenic therapies, and the very cell you're trying to kill evolved to live in hypoxia. It's not going to work, and it doesn't work. In fact, some of the leaders come out and said, yeah, this stuff isn't working. All right, so what do? What do they eat? That's what you have to know. What are these cells eating? They can't live without energy. Okay, so they eat glutamine. We know that. Our immune system is driven by glutamine and glucose. So if most cancer cells have tended to fermentation, how do we manage this stuff? One strategy is to take away their fermentable fuels. Not complicated. Okay, so what did we try? We tried calorie restriction, fasting, ketogenic diets, this kind of stuff. What it does is lowers blood sugar. It creates a system of starvation, but it's not starvation. It maintains minerals and nutrients. You can lower the fermentable fuel glucose and elevate the non-fermentable ketone bodies. Tumor cells can't use ketone bodies because their mitochondria are shot. You also get biogenesis of mitochondria. You can help, the normal cells get really healthy. The tumor cells start to get hammered. And water-only fasting is the way to do this in humans, calorie restriction in mouse. So that's why we went into ketogenic diets, because nobody wants to fast. You try doing it, it's not easy. So what are these ketogenic diets? A lot of talk about this. Most people using it for weight loss or whatever. We're using it as a medicine. We use it as a medicine to kill cancer cells. Okay, so it's a low-carb, high-fat, moderate protein diet, but it's not that simple. The types of carbs, fats, and proteins and their ratios play an extremely important role. This is not an Atkins diet, okay? You got to be able to lower that blood sugar if you want to kill these tumor cells. You can't eat a lot of protein that raises blood sugar, so it doesn't work if you don't do it right. So that's why I say on the bottom, it's got to be restricted, because if you eat too much of it, it doesn't work. So people need to know that. And what we did is we published a lot of papers showing that if you can lower the blood sugar, which is the dark line, and raise the ketones, you can then transition the entire body over to a new homeostatic state. So the ketone bodies are going to serve as an alternative fuel to glucose, especially for our brain. Our brains are driven by glucose, but we evolved to be able to burn ketones as an alternative to glucose. So by lowering glucose, we put pressure on the fermented capability of the tumor cell. And raising ketones, we're now enhancing the health and vitality of normal cells. And we can then use drugs to push blood sugars down to really, really low levels, because everything is transitioned over to the alternative fuel. So once we can get these people, mice or patients, into this managed growth, the tumors now become vulnerable and the normal cells become protected. And that's the strategy that we're using to manage the disease. Now, the first person to do this, one of the first persons, but Linda Nebling took two little girls, hopeless cases, at Case Western Reserve, and treated them with ketogenic diets when they were given up as hopeless by the medical establishment at that time. She lowered blood sugar, raised ketones. Both of these hopeless kids responded remarkably well. You should read the paper. Their quality of life improved significantly, and their overall survival increased dramatically. So I looked at this and I go, my God, what's going on here? I got to see what I got to do with this on a mouse. And we did it on a mouse. So this is a very aggressive CT2A malignant astrocytoma, like a glioblastoma, and I'll talk more about that. So we just simply lowered blood sugar by giving them a 40% restriction. Standard diet is a high carbohydrate diet, unrestricted food. Same diet, restricted to reduce 40%. And we got anywhere from a 65 to an 85% reduction in the size of the tumor just by cutting back the calories. So what's going on here? So each one of these squares is a mouse under a different dietary condition. We did linear regression analysis. Okay, as the blood sugar goes down, ketones go up. It's an evolutionarily conserved adaptation to fasting. As blood sugar goes down, the size of the tumor went down. The higher the sugar, the faster the tumor grows. The lower the sugar, the slower the tumor grows. This has been repeated in humans. A number of cases in humans have been shown. The higher you want your tumor to grow fast, make sure your blood sugar is as high as you can get it. Chug that high fructose corn syrup, right? You go to oncology clinics to get ice cream, Enfamil, all this kind of stuff. This is published in the literature. Let me read this stuff. Anyway, what's the mechanism by which it works? Oh, we published the mechanism. People, like we heard from Dr. Calabrese, you have to have mechanisms. We published the mechanism. Anti-angiogenic, anti-inflammatory, pro-apoptotic. There's no cancer drug that can do this. That's been published again and again by so many people. What's the mechanism? What's the mechanism? Here's the mechanism. Now I'm going to talk a little bit about glioblastoma. As we know, Senator McCain is now struggling with this, and we know of many other people in the past who've unfortunately had to struggle with this. It's a very aggressive tumor, all dysmorphic kinds of cells. The tumor cells invade throughout the brain using secondary structures of Schur, can metastasize out. There's a very difficult, and I'll show you the data. Okay, so here's a section, GBM. You can see the midline shift, the crushing. Most of these poor people, they die from intracranial pressure from the expanding tumor, the edema, and everything else that goes on. The right here, the blue cells, what it's showing you is cancer cells migrating through the brain using on the Virchow-Robin space. It's the space on top of the blood on top of the blood vessels. There's a little space, and these tumor cells will run. They'll use these blood vessels as a railroad system to get through your brain. So even if you try to cut out the center part of the tumor, bang, these guys are out. So this is what makes it so difficult. And this is why some of these say, oh, we're going to do immunotherapy. They're on the other side of the blood vessel. They're not going to be touched by the immunotherapy for the most part. So it's not going to work. Somebody, you might find a person here and there, but basically it's not going to work for the majority of people, no matter what people say. Now, what's going on in the mitochondria of these tumor cells? So I have here the normal mitochondria. You see the stripes, those are called cristae. They contain the proteins and the lipids of the electron transport chain, which give these cells an opportunity to respire. And you see the glioblastoma mitochondria right there. It's crystallisis. These are ghost mitochondria. You find all kinds of weird things on the mitochondria, all kinds of tumors, not just glioblastoma. So if those stripes and those cristae contain the very molecules necessary for generating energy through oxidative phosphorylation, and those structures are not there, what do you think the probability is of that organelle producing energy through oxphos if the very structures needed aren't there? Makes sense. They can't do it, right? That's why they're fermenting and blowing out massive amounts of lactic acid. Okay, we have seen this over and over and over again on tissues from humans and mice and rats, damage to the mitochondria structure and function, and the field ignores it. They say, oh, no, cancer cells have normal respiration. It's unbelievable. So now what's driving the beast? As I said, fermentation drives the beast. Okay, comes glucose and glutamine. Glucose come pouring into these cells through an upregulation of glucose transporters. The glucose is metabolized through the Embden-Meyerhof pathway to pyruvate, which is then dumped outside into the microenvironment, acidifying one of the many molecules. It also goes down the PPP as the pentose phosphate pathway, producing precursors for nucleotide synthesis so the cells can grow. And they also produce a lot of glutathione, which protects them against oxidative stress. Very important to know that. And then we have on the bottom, we have the glutamine pouring into these cells. The amide nitrogen is used to make proteins and nucleotides. The glutamate then enters, either dumped into the microenvironment to facilitate the invasion, also goes into the mitochondria, converted to alpha-ketoglutarate, and generates a massive amount of energy through substrate-level phosphorylation at the succinyl-CoA ligase step, which Warburg didn't know about. Okay, that's the missing link in Warburg's theory. These cells are fermenting, and it makes it look like the mitochondria are working, generating ATP, but it's not coming through oxphos. So let me show you what we do to the people in the clinic. I published this paper in a top journal, Lancet Oncology. So person's diagnosed with GBM. It's unbelievable. You know, it's shocking when you get that kind of a diagnosis. It's just brutal. So before the family or the patient really understands what's going on, you know, they have you taking the tumor out right away. And that creates a necrotic microenvironment, because they always say you can never get it all. These GBMs are rarely debulked 100%. There's always a piece or some part remaining. And the damage that you've done to the microenvironment from the surgical debulking will facilitate the return. And then we start to irradiate as those bolts of light, the bolts up there. Okay, creates more edema, creates necrosis. All that's freeing up glutamine, breaking the glutamine-glutamate cycle, very tightly regulated cycle in the brain for those of you who do neurobiology. Okay, we're freeing up the very molecules that are going to be driving the beast. And because the cells, the head starts to swell, the swelling, the edema, we give them high-dose dexamethasone, right? To lower the swelling and the edema, and that raises blood sugar. All right, so what are we doing here to these poor people? We're creating a microenvironment that's going to guarantee the recurrence of this tumor and the likely demise of the patient. And many of these tumor cells are infected with human cytomegalovirus, which acts as a supercharger of bringing glucose and glutamine into the proliferating tumor cells. So what do you think the outcome is going to be on patient survival from the standard of care? And these are the data published over and over and over again. All right? Look at that red line. Those are the people that were irradiated. No survivors. And this is done over and over again. And then they add the toxic temozolomide in there, and that was the first major breakthrough in glioblastoma, brain cancer. They live a little bit longer from a toxic drug. That also increases driver mutations. Try to figure that out. These things make the patients get sick. They have diarrhea, vomiting, nausea. All these are indirect forms of calorie restriction. It's not working. Okay, now I'm going to talk about individuals. Remember Brittany Maynard. It's a big story in People magazine and everything a few years ago. All right, diagnosed with a low-grade tumor. She was 27. All right, low grade. Okay, and within a month, it ripped into a GBM because you poked around. It's evidence now that we poke into these tumors, you can explode them. This is her picture right after she got married. So what happened now is she goes on the standard of care. You can see her on the right here with the swollen face, moon face from high-dose steroids. Doesn't even look the same. And then there was a big article in People magazine, My Decision to Die. She doesn't want to go through the standard of care anymore. She doesn't want to do this. She's going to throw the towel in. Made a big, she was from California. She just had to go to Oregon. They have the Death with Dignity thing up there. So she dies with dignity on November 1st, 2014. And the big article in People magazine was, oh, is it morally right to take your own life with death with dignity? Not the fact that the standard of care put her in that place in the first place. There was no discussion about the failure of the standard of care on that article. Read it. It's People magazine. Now, this is another case, Pablo Kelly. He diagnosed with a glioblastoma in 2014, same year as Brittany. He contacted me and he said, what should I do? I said, I don't know. I didn't tell him don't do standard of care. He came to that decision on his own. He told those guys, well, you're going to be dead in 3 months, Pablo. 3 or 4 months, you're going to be dead. He said, I'm doing ketogenic diet. I said, well, good luck. I don't know what to say. You know, it was very early in our experiments. We didn't know. So I hadn't heard from Pablo in, I don't know, a year and a half. I said, maybe the guy's dead. I don't know. All of a sudden, this paper, Herald Express in British, South Devon cancer sufferer Pablo defies doctor with special diet. I just emailed him 2 weeks ago. I said, Pablo, how you doing here? Still surviving, 2018. Rejected the standard of care. He's not alone now. There's more and more people that are rejecting the standard of care because they want to live. And that's what Pablo said, I want to live. Okay, I'm going to meet him at a meeting in London next fall. He's going to be one of the keynote speakers, going to tell everybody how he survived. He's going to be 4 years. You know how rare it is to get to be 4-year survivor with a glioblastoma? Not too many people in that category. So the problem is we have a lot of patients, they don't know what to do. The physicians don't know what to do, and the patients don't know what to do. How do you know, how do you implement this stuff? Okay, I can tell you all this stuff, but I have a tumor, what am I going to do? So my students and I came up with this glucose ketone index calculator. All it does is calculate the ratio in millimolar of glucose to ketones. And we predicted that maybe if you can get your ratio to 1.0 or below, you bring in your body into this new metabolic state that's going to put pressure on the dividing cells, and maybe that can keep you alive a little bit longer and a higher quality of life. So we've been doing that, and people are now feeding us back information saying, yes, if I get into that zone, I can hold down the beast. I'm not saying we cure the cancer, but I'm saying we can hold it down. We can slow it down. Well, if you can slow it down, then you have opportunities to attack from other angles, and that's what I'll show you what we do. So what we did then with my colleagues, George Yu, who's an oncologist from Annapolis, Maryland, Joe Maroon, who is the team surgeon for the Pittsburgh Steelers, distinguished neurosurgeon, Dom D'Agostino from South Florida, myself, we put together this press pulse concept to treat cancer, not only GBM, but all cancers. And you can see the red, we want to take the sick patient, we want to first of all try to manage it as best we can, and then we go for the resolution. All right, so we have to slow down the growth, and we do that. Now this concept of press pulse came from paleobiology, if you can believe it. It turns out that in the past history of the Earth, there was massive extinctions that occurred when two unlikely events happened at the same time. Okay, a chronic stress on a population coupled with a massive, like a meteor hit or some volcanic explosion or whatever, wiped out everything. So we took that concept of paleobiology and brought it into the field of oncology. And to do it, the key is to do this without harming the normal cells of the body. This is the strategy. You've got to be able to degrade and eliminate the tumor without killing or harming any of the normal cells in the body. And this press pulse concept is designed to do that. So we get the patients into the blood glucose ketone zone. We use stress management because people are all freaked out by having cancer anyway. They get freaked out, their blood cortisol goes up, high glucose. So you got to use all kinds of stress management techniques, exercise and things. And then we hit them with pulses. Doses, timing, and scheduling of drugs that target glucose and glutamine. And we use hyperbaric oxygen. And now I have plans this summer to do ozone therapy. I haven't done ozone therapy yet, but from what I heard today and what I've heard from others, it sounds like it's perfect for this. Combine ketogenic diets with ozone therapy together with these other strategies. I haven't done it yet, so I can't say anything about it. But I had the plan. I have a plan. There's a veterinarian who wants to work with me. So I said, okay, we'll give it a shot. And it might be easier than hyperbaric oxygen. Why we do hyperbaric oxygen and potentially ozone? To replace radiation therapy, because both of them are going to kill the tumor cell through oxidative stress. But we want to do it so not to harm the normal cells. So doses, timing, and scheduling, in my mind, and certain time, and I have to be honest with you, we're not there yet. We're still fumbling around with this. But the strategy is there. And I think in time, this strategy at Press Pulse will make most other cancer therapies obsolete. Just a matter of time. Now, how did we do this? We just didn't pull it out of the air. We developed a tool. We developed natural models of glioblastoma, spontaneous in the brain, not these genetically created things that you see and you hear about. These are natural things. If you want to make an advance, you better make sure you have the right tool. So I said, let's try to kill this. This is a VM glioblastoma model. It has all the characteristics of you. We published this, you can see the papers. So here's one of our attempts. Okay, you can see the tumor in the brain. Here's the hippocampus. And the all the high carbohydrate, standard high carb diet, tumors ripping right through the brain using those Virchow-Robin spaces, doing all this. The tumor on the right, calorie restriction, ketogenic diets. The invasion was much less, but we weren't killing all the tumor cells. What's going on? I threw everything at but I can't believe what I did to these poor mice to stop that tumor. I couldn't stop it with targeting glucose. So what are they living on? What do they got to eat? They got to live, right? I said, let's try the glutamine thing. So we used Don, an old drug, unpatentable, that was used years ago in the oncology field. They don't use it anymore because they didn't know how to use it. It wasn't used correctly, but it worked on a number of people. So we said, try it because we need to test the hypothesis. Is it glutamine that's keeping these guys alive? So it targets glutaminase, so it doesn't let the glutamine anywhere into the cell. It basically stops the glutaminase, so you don't get the glutamate or the amide nitrogen. So we put the tumor cells into the mouse's brain, wait 3 days until it's a raging tumor. And then we started the diet. We had two diets, the high carb and the ketogenic restricted as the press. And then we pulse, bing, bing, every couple of days we hit him with the Don. Now we stopped the experiment at 15 days because the guys eating the high carb diets are going out on us, right? Becoming moribund. So, but the other guys getting the press pulse are looking really good. Okay, they're looking, wow, they're upset that we have to stop the experiment because they have to sacrifice their life so we can look and see what's going on. Okay, so the cells are engineered to be bioluminescent, so we can take and quantify the degree of tumor growth by looking at bioluminescence imaging. So on the top is you have to put sunglasses on. Some of the tumors are raging hot from the high carbohydrate diet. The ketogenic diet restricted has a little bit of an effect. I told you we couldn't stop these things. Don, I don't have the Don. Don by itself was a little bit better than the ketogenic diet restricted, but there was still a lot of hot tumor cells. When you put the diet and the drug together, there was no light in the brain. There was no light. I said, what happened? What happened? So we then looked at individual mice. These are each number is a mouse under a single mouse. So the ketogenic diet, everybody had a light in the brain. That means we weren't able to kill and eliminate this tumor with ketogenic diet restricted by itself. But when you put the diet and the Don together, those little blurbs at the bottom are background. And there was one guy, there was number 6 there. He had a little bit of a light, only because we backed off a couple of injections on him. But we knew we could explain why he broke through. But all the others were pretty much no light. The other thing that was spectacular was that by administering the drug in the ketogenic diet, we could deliver twice as much drug on target than when we could deliver the drug with a high carbohydrate. So whatever's going on here, ketogenic diets are facilitating the delivery of the drug to the target. So that means we can use less concentration of the drug, which is great. Then we said, what's going on inside the cells of this guy's brain, these mice? So here's the histology, low power 100x, high power 200x. So standard diet, unrestricted, you can see the dark blue is where the heavy tumor cells are, and the lighter area is the normal brain. And you can see the massive invasion of the cells into the normal brain. These cells are fat and happy, growing like crazy, many mitotic figures in the high carbohydrate diet. Ketogenic diet restricted, you can see the white part. There's a demarcation between the tumor and the normal brain, and we're stopping the invasion. Yes, the ketogenic diet is reducing the invasion. It's also causing more spacing. The cells are not dividing quite as fast. But then when we put the diet with the Don, we slaughtered these things. This is all dead cells. That's all necrotic death. Mitotic catastrophe, necrotic death, all different kinds of death. It's a battlefield. We slaughtered those tumor cells. Combining the diet, we're targeting the glucose and we're targeting the glutamine simultaneously. Just to show a quick picture, this is my colleagues Dominic DeAugustina, Angela Poff from University of South Florida. We put the cells into the flank of the mice, and here's where we used hyperbaric oxygen together with the ketogenic diet, and we got significant synergy. And as I said, maybe ozone would do the same thing. We haven't tested it yet, but we're going to be we're building cocktails, we're building all these things now. Now, how does it work in humans, besides what Linda Nebling had shown? Okay, this was our study that we had to do in Istanbul, Turkey. These guys that read my book, they said, oh my God, we should be using this kind of a strategy. So they're treating all kinds of stage 4 cancer patients in Istanbul, in Turkey. Really pancreatic cancer, breast cancer. This is a woman with triple negative breast cancer. The arrow number 1 is pointing to the breast where the tumor is. It's in the liver. It's in certain muscle sets. It's metastasized. They put her on the call it metabolically supported chemo, the lowest dose of chemo, used with hyperbaric oxygen, used with ketogenic diets, insulin potentiation, little metformin. So they're building these cocktails, and you can see on the right, the woman's health is really good. Her quality of life is excellent. She's experienced no adverse effects from the therapy at all. And they even told me, we have to use the chemo because they're going to take our license away if we don't throw some chemo. We use the lowest dose. I said, what would happen if you use no chemo? That should be better. And we're getting the same thing with pancreatic cancer. You can't believe it. Ovarian cancer, non-small cell lung cancer. So they have a big clinic over there, and they're being overwhelmed. Now, you don't have this picture. This paper just came out this week. It's not even out yet. This is the abstract. It's our paper that I worked with guys in Alexandria, Egypt, that had a guy with a brain tumor, young man, 38 years old. I had no idea that they were doing this stuff. They contact me and they say, oh, you got to see how well our patients are doing using metabolic therapy on the cancer. So I said, tell me about it. Because, a lot of these physicians, they're great, they're wonderful. They don't write papers. They just run around telling me, hey, that works. Where's the evidence? Where's the literature? So I said, well, we have to publish this. I worked for over a year on this with my colleagues, Perna and Mariam Kalamian, who wrote the book Keto for Cancer. Excellent book. You should read that book. Mariam Kalamian, Keto for Cancer. Anyway, she worked the diets. This guy was in bad shape. He came into the clinic. He had deficiency in vitamin D. He had triglyceridemia, his full triglyceride. His whole body was out of whack. And beside, he had a glioblastoma. His whole left side is paralyzed. So they put him on. Now, what we did differently here, and that we've never done anywhere for the first time, is we instituted metabolic therapy before standard of care. Okay, so he went on a 3-day water-only fast, transitioned to a 950 kilocalorie a day ketogenic diet for 3 weeks, and then they did the bulking, and then we went through with the standard of care, together with a modified ketogenic diet, chloroquine to stop glutamine. We used EGCG, which also targets glutamine. We did hyperbaric oxygen. In other words, we threw all the things that we thought would stop glucose and glutamine. The guy's doing fine. He was a farmer, a corn farmer. He's back out in the fields as we speak, right? He's out 25 months now, and he's out working in the fields. I don't know how long the guy's going to live. All I know is he's not suffering. All right? He's not suffering. So he's doing well. So we're just going to keep going forward with these kinds of experiments and see what happens. How many people can we keep alive in a quality of life? That's the key. All right. Conclusions. Cancer is a type of mitochondrial metabolic disease. It is not a genetic disease. Okay, and I think this unfortunate misunderstanding will at some point be recognized as the single greatest tragedy in the history of medicine. That we have misunderstood a disease that has led to the suffering and death of millions of people around the world because of a misunderstanding. A reliance on substrate-level phosphorylation for energy is the metabolic hallmark of nearly all cancers. I say nearly all because as far as I can see, they're all. There might be one out there that's not. The simultaneous restriction of glucose and glutamine, together with what I've shown you, can manage GBM and most other cancers. The press pulse metabolic therapy is non-toxic, cost-effective strategy for the management of and possible resolution of all types of cancer. Cancer is not 100 different diseases that the NIH says it is. It's one disease of energy metabolism, and that defect is in the mitochondria, and it's causing all these other repercussions. And all this crap that we're doing in the field is most of it is a waste of time and money. So my collaborators are in the United States. We see a bunch of guys that I've been working with for a long time. Now we're collecting collaborators throughout the world, in Turkey, Germany, Venezuela, Hungary, Greece, France, Egypt. I just came back from Shanghai, China. They're going to try to dovetail our metabolic therapy in with their traditional Chinese medicine, and they're very excited about this, and they've got many, many patients lined up to try to do this. I also want to thank my funding agency, Single Cause, Single Cure Foundation, Travis Christofferson's foundation, George Hughes Foundation, Joe Mercola, Ellen Davis, Ed Miller, Boston College Research, and in the past, the NIH. All right, it'd be wonderful to get money from them, but it's hard to get money when you're doing therapies that actually might work. Thank you very much for your attention.